Hello, students, and this is Professor Chakraborty. Today we are going to learn about neurons. We'll start with cells in general, all cells in the body, and then gradually go into the neuronal cell. Cells are the fundamental building blocks of life, performing structural, functional, and biological basis of all living organisms. As you can see in this figure, that from embryological perspective, all cells originate from one single cell. All cells, whether they are multicellular organisms or a single cell organism, they all originate from a single fertilized egg, which is the zygote, and undergoes repeated mitosis in order to form different types of tissues. Cells into tissues, tissues into organs. So you can see it in figure that these cells are differentiated into different kinds of tissues. As embryogenesis progresses, these initially identified cells differentiate through regulated gene expression, giving rise to specialized cell types that form tissues and organs. Now, despite their structural and functional diversity, right? They're pretty different in their structures and their functions. All cells retain the same genetic blueprint, underscoring or highlighting the unity of cellular origin and development from a single cell. This embryological continuity shows how complex organisms arise from a single cellular beginnings and undergo growth, differentiation, and organization in order to form an individual. Going forward, you must have learned this earlier in your earlier different classes. We're talking about the cell theory. In 1665, Robert Hooke first described the name cells after observing cork under a microscope, although he was viewing dead plant cell walls. To him, these cells looked like cells of the jail, and that's the reason he called them cells. Later, in 1830, Matthias Schneider proposed that all plants are composed of cell, and Theodor Schwann later proposed the idea that all animals also follow the cell theory, that they origin from cells. Later, Rudolf Virchow added a very important principle, which is called omnis cellula e cellula. That means all cells arise from pre-existing cells, emphasizing cell division as a basis of growth and development. Collectively, these discoveries from these eminent scientists led to the cell theory, which is the building block or foundation of modern cell biology, embryology, and medicine. So, cell theory is one of the foundational principles of biology. It states that all living organisms are composed of cells, and cells are the basic structural and functional unit of life. There are three core principles of cell theory. Number one, all living organisms are made up of one or more cells. This applies to unicellular organism. It applies to multicellular organisms. The cell is a basic unit of structure and function. That means all physiological functions that occur within the cell, that is, for example, metabolism, energy production, growth, everything happens within the cell. Third, all cells arise from pre-existing cell, which was basically the foundation of Rudolf Virchow. Omnis cellula is cellular, that they arise from the pre-existing cells. This cell theory is universally accepted for all cells. Going forward, in order to implement the cell theory into the nervous system, it was found that by the initial scientists that the cell theory did not work for the nervous system. That's what they thought. It was initially done by two eminent scientists. Two eminent scientists in here we are talking about. One is Camillo Golgi and Santiago Cajal. These are the two main scientists. There were many, many other scientists who had an equally important role, but these two scientists are considered to be the most prominent scientists in order to design the cell theory for neurons, which is known as the neuron doctrine. It is important that to understand that Camillo Golgi, who was a prominent scientist, developed a stain which is known as the Golgi stain. And this stain actually revolutionized the entire understanding of the neurons. So, what are neurons? Neurons are the structural and functional unit of the nervous system. This stain, the Golgi stain, was able to differentiate between the cell body and the neurites. What are neurites? Neurites are the extensions that come out of the cell body. We'll learn that eventually that these neurites are nothing but the dendrites and the axons. This Golgi stain was able to differentiate between the cell body and the neurites. And he proposed a theory which is known as the reticular theory, where he said that these cells are continuous, that is, one cell communicates with the next cell continuously. They are in contact with each other, they touch each other. And that's what the reticular theory that was designed by Camillo Golgi. Later, Santiago Cajal, using the same stain, that is a Golgi stain, and further using Nissl stain, proposed that the cell and the neurites are not continuous. That means the neurites do not touch another neuron's neurite, which is fundamental and the basis of the neuron doctrine, that the cell of one, that is one neuronal cell, and the next neuronal cell do not touch each other, and there is a gap in between, which is known as synapse. This led to the theory of which is known as the neuron doctrine, which was mainly proposed by Santiago Ramon Cajal. These two gentlemen, although they had different theories, but the basis of their work was so important, and because Golgi stain helped in designing the neuronal connectivity, understanding of the neuronal connectivity, both these gentlemen shared the Nobel Prize, although they never agreed upon their theories, indicating a very, very important part in the scientific world, that the shared Nobel Prize highlights how scientific progress often emerges from debate and contrasting interpretation, with Kayal's neuron doctrine ultimately becoming the accepted model of nervous system organization. So, what are the core principles of the neuron doctrine? Number one, neurons are individual cells. They are structural and functional unit of the nervous system. Neurons are not physically continuous with one another. They they're not joined to each other. So they communicate with one another through a junction which is known as synapse or synaptic cleft. Neurons communicate at specialized junctions. These contact points are synapses, which I just said. Transmission can be electrical or chemical. Then a very important part, neuron polarity, which differentiates neurons from rest of the cells. Dynamic polarization, that is, it can be excitable and the flow of information is in one direction. That is, from dendrites, it goes into soma, it goes into axon, synapse, and then it passes on to the next neuron. One directional information or electrical transmission occurs from one cell to the other. Connection specificity. Neurons form precise and highly organized connection. We'll learn that in next slide. How neurons form a network in connectivity, and that is very important in order for us to function, in milliseconds. Each neuron connects only to specific target cells, forms accurate neural circuits. Neurons are embryological derived units. Neuron has a distinct cell that develops independently during embryogenesis. So the neuron doctrine is the fundamentally accepted foundational concept in neuroscience, stating that the nervous system is made up of discrete individual cells called neurons rather than a continuous network, and it also follows the cell theory. The connectivity that we were just talking about is at a very high level in case of neurons. There's a molecular level, cellular level, synaptic level, microcircuits, pathways, tracks, interregional circuits, systems level, behavior and cognition. All of this we will be covering in this course. This is a general structure of a cell. So if you look at it, you will understand that this outer membrane is a plasma membrane and it is a selective permeable membrane. It is made up of phospholipid bilayer. And inside the cell, which is made up of cytoplasm, there are several organelles inside it: nucleus, rough endoplasmic reticulum, Golgi apparatus, mitochondria, lysosome, centriole. I repeat again, this is a generalized cell. Any somatic cell we are talking about. We are not talking about neurons here. Any cell in your body has all these organelles as shown here. In case of neurons, we'll learn at length. The structure, this is the structure of a neuron. It consists of three major parts. Number one is a cell body. It has extensions that are coming out, which are known as dendrites. And then a slender process, which is known as the axon. And then it ends into telodendron or axonal terminal. Three major parts: dendrites, cell body, and axon. Within them, there are certain subparts which we will learn later. The cell body is also known as soma, and this cell body contains all the organelles that any other somatic cell carries, except for one, which is the centriole, and we'll learn that eventually. So, in order to learn a more detailed structure of the neurons, again, this is the cell body, these are the dendrites, and this is the axon, this is the axonal terminal. The dendrites have spines, dendritic branches, dendritic spines. And in the cell body, there are all the organelles that are required, the main function being protein synthesis. So it has the nucleus, which carries DNA, the blueprint for all the proteins. The DNA is converted into mRNA and then into proteins by the rough endoplasmic reticulum. The rough endoplasmic reticulum, as you can see, this is the endoplasmic reticulum and it is studded. So there are the rough endoplasmic reticulum. The rough endoplasmic reticulum in case of neurons is also known as the Nissl body. So going forward, whenever we refer to rough endoplasmic reticulum in neurons, we will be calling it Nissl bodies. So in the DNA forms mRNA, mRNA forms protein. So what kind of proteins are these neurons making? There are several proteins that are required by the neurons to function. There are ligand-gated channels, which are made up of proteins. There are sodium-potassium pumps, which are made by the cell body. There are several enzymes that are required. There are certain, we will learn in the axons, there are transport systems that are made up of dynein and kinins. Those are all proteins that are made by the cell body. So these proteins are made in the cell body and they then are passed on into different regions of the neuron for their function. So this is the cell body. And then these are the dendrites. These are the dendritic spines. And then comes the axon. The axon, the initial part, the part where the cell body tapers, is known as the axon hillock, a very important region. It's called the trigger zone because this is where the initiation of action potential takes place. What is action potential? How it works? We have a separate lecture on that. So once that is initiated, it leads to the axon. The axon can be either myelinated or maybe unmyelinated. What does it mean? Means that it can be covered by a lipid layer made by a neuroglial cells. What are neuroglial cells? These are supporting cells which help in the neuronal transmission. So if they are, covered by this lipid layer made by these neuroglial cells, which are known as oligodendrocytes or swan cells in the PNS, they are myelinated. And if they're not covered, they are unmyelinated axons. And then leads to the last part, which is the axonal terminal. This is where there are tons of vesicles, neurotransmitters. These neurotransmitters are made in the cell body and transported here for the function. I want to mention here one very important thing is that each neuron actually acts as a presynaptic neuron and a postsynaptic neuron. Then comes, in order to understand the function of the neurons, this is the receptive end of the neuron. This is the conducting region, which is the axon mainly, and this is the secretory region. Receptive neuron is these dendrites. It's like here in your body, which takes in information. Receiving end takes in information, and then it processes it in the cell body and then conducts it in the axons. Through the axon, it can be myelinated or unmyelinated, and then passes out to the next region, which is the axonal end, which is the synaptic end, and then it passes the information from the synaptic end to another neuron. So these are the different regions of the neuron. We'll learn about all of it in details again, but I just want to go over the different parts of it. There are dendrites, the soma, the cell body, axon hillock, axons, nodes of Ranvier, myelinated segment, and axon terminal. We'll do the rest a little later. So we are learning about each part of the neuron. First, the dendrites. As you can see it in here, the dendrites, this is the synaptic cleft. So this is the axonal end. This is an axon. Till here is the axon. This is the axonal terminal. In here, there are several vesicles called neurotransmitters. And these vesicles are being transported or released out from the axonal terminal into this cleft, which is known as the synaptic cleft, which is taken up by the postsynaptic neuron. In the postsynaptic neuron, there are receptors. There are receptors. When these neurotransmitters are released, they are taken up by these target receptors, and the action takes place. So these synaptic terminal is releasing these neurotransmitters into the postsynaptic neuron, which is taken up by another dendrite. This is a dendrite of the another neuron. So if you look in here, you'll understand it better that this is a neuron. These are the dendrites. This is a cell body. This is an axon. This is the axon terminal. So these dendrites are taking up information from the axonal terminal of another neuron. So the process of synapse, taking up, dendrites are taking up information, processing it, and then transferring it to the axonal terminal. Here again, it is being taken up by another neuron, which is nothing but synapse through the dendrites. This is the presynaptic terminal. This is the postsynaptic terminal. This is the presynaptic terminal is axonal end, and the postsynaptic neuron is the dendrite in here. So the process actually transfers the neurotransmitter signals from one neuron through the action potential, goes to the next neuron, and so on and so forth. Let's understand this again very well. Now, if you look in here, you will see this is a neuron, of course, but this is the axonal end of a neuron. And this is another neuron, and this is a dendrite of another neuron. In the axonal end, there are vesicles which diffuse out into the synaptic cleft. And these vesicles carry neurotransmitters, these neurotransmitters which are made in the cell body. In the postsynaptic neuron, which are the dendrites of the postsynaptic neuron, there are receptors waiting for this ligand or neurotransmitter to get attached and to transfer the information. Now, this process can be of two types. One, it can be EPSP, which is excitatory postsynaptic potential, IPSP, which is inhibitory postsynaptic potential. So these neurotransmitters can be excitatory, that means it can excite the next neuron, or it can be inhibitory, that means it can inhibit the rest of the process. There are other processes also, which is G protein coupled receptor, second messenger, which we will not discuss in here. So the dendrites have a very important function. They are the receiving end. They take in information, which is processed by the neuron, by the cell body particularly, and then they transfer through the axon to the next neuron. So what are the general functions of the dendrites? They receive synaptic inputs from other neurons, as we just learned, integrate excitatory postsynaptic potential and inhibitory postsynaptic potential to determine neuronal output. Neuronal output means how the neuron is going to act, whether it will excite another neuron or it will inhibit another neuron, depends upon these information. Conduct graded potentials towards the soma. Very important. What is graded potential? We'll learn that again later. But remember, graded potential is an initiation of an action potential, which means how the neuron is going to transfer electrical impulse through the neuron depends upon how the dendrites will conduct the graded potential. Increase surface area for connectivity and information processing. It has spines, primary spines, secondary spines, tertiary spines, and increases the surface area so that it can take information from multiple neurons or neuron axonal ends, support synaptic plasticity through dendritic spines, enable compartmental processes within different branches. So where each information will go into which region is also determined by the dendrite. The cell body. The cell body is interesting. The cell body has very, very similar composition as that of any other cell. It has a nucleus. It has rough endoplasmic reticulum, which is known as the Nissl bodies. These are ribosomes studded with endoplasmic reticulum, rough endoplasmic reticulum. There are Golgi apparatus. There is mitochondria, which is very important for neuronal function. There are pre-ribosomes, and there is another factor, which are neurofilament and microtubules. Very important. Microtubules, neurofilaments. Neurofilament actually gives neuron a structure. Microtubules are present, which actually help stabilize the transport system of the impulses. We'll do that eventually. You will see that how information can transfer from one region to another. But in addition to that, the proteins that are made in the cell body needs to be transferred from the cell body to the axonal end, or need to transfer from the axonal end to the cell body. Those railway tracks, you can say, or you can say those are proteins that help in the transfer of proteins, are actually stabilized by these microtubules. Disarray of these neurofilaments or microtubules can lead to a very significant and well-known disease, which is known as the Alzheimer's disease. That's microtubule associated protein known as tau, and that actually accumulates in such a way that the transmission is blocked. So it is important to remember that these neurofilament and microtubules play a very important part in the neurons. So what are the functions of the soma or the cell body? It is a metabolic center of a neuron, contains nucleus, Golgi, mitochondria, which is endoplasmic reticulum, and ribosomes. They are the main region for protein synthesis, enzymes, and neurotransmitters. This is the reason why it is so important, because neurons cannot divide. I go back one slide to make you understand that there are all these organelles present, but one organelle is absent in case of neurons, and that's the centriole. The centriole is not present, and centriole is responsible for cell division. So as a result, the repair mechanism of the neuron is relatively difficult, and in neurons heavily rely on the soma to maintain a cellular health. Integration of incoming signals. So we learned that the dendrites bring in the EPSP and IPSPs, but how they take in these information and make it into graded potential, which eventually will become an action potential in the axon hillock, happens in the cell body. So the integration part happens in the cell body, maintains neuronal structure and function. We just learned that, then microtubules, neurofilaments are there. In addition, it provides ATP because mitochondria is present in there. It synthesizes proteins for dendrites and axons, supports repair and regeneration in the PNS. Just to make you aware, it's not CNS, it's PNS. The repair mechanism of CNS is relatively poor in case of neurons. Produces neurotransmitter precursors, Enzymes required for neurotransmitter synthesis are made in the soma and then transported into the axonal end. Support axonal transport, they actually make the proteins that help in these transport, which is kinesin and dynein. We'll learn about anterograde and retrograde transport very soon. The third part is the axon. So if you see, this is the soma, these are the dendrites, these are the dendritic spines. In the soma, this is the nucleus, and the point where it tapers is known as the axon hillock. That's the beginning of the axon. Remember, this is a very important region because this is a region where the action potential is starts or is created. Then axon proper, which is in this region in here, and then the terminal. Now, what is the difference between axons and soma? Endoplasmic reticulum does not extend into the axon, and the protein composition of each one is different. Axonal function, it conducts action potential. 5 stars for this function because this is the major function of a neuron. Because it is responsible for action potential, and that is mainly responsible for conduction of impulse. Transmits signals rapidly, saltatory conduction in myelinated axon. So you will see, we'll learn about saltatory and continuous conduction later on, just to make you understand that once the neurons take in information, they have to be transported. It can be transported very fast in myelinated neurons. Myelinated axons means these are axons that are covered by a lipid layer made by oligodendrocytes in CNS and Schwann cells in PNS. And this rapid transmission is required for all actions in our body. This rapid transport is only possible because the axons mainly are myelinated. Release of neurotransmitters in the axonal terminal. There are vesicles that can actually, that are responsible for release of neurotransmitters from the axonal terminal. And this release of the neurotransmitters is essential because that's the next step, that is transmission from one neuron to the next neuron. So the very important that it transfers these neurotransmitters into the next neuron. Carries material via axonal transport. So the rail tracts that the proteins are made in the soma are transported through the rail tracts through anterograde, and they are brought back to the soma through the retrograde, forms axon branches to influence multiple targets. There are even collateral branches of the axons so that more and more information can come in. Supports neuronal survivability and plasticity through transfer of tropic and signaling molecules. So it is important that this regeneration of the axons is part of the axonal function, and it is how diverse the neurons are able to function depends upon the axons as well. Okay, so now we want to understand one basic information which we all know from our previous knowledge, which is all cells are made up of plasma membrane. So if we if you remember that the plasma membrane covers the entire cell, any cell, and it is made up of its phospholipid bilayer. and it is selectively permeable, that it allows certain chemicals to come in, certain chemicals to go out. It is selectively permeable, and it is plays a dynamic role in cellular activity and has many glycoproteins on the surface of the cell. So if you look at the picture, you should be able to understand this better. This is a phospholipid bilayer. There are several channels, glycolipids, glycoproteins attached to it. So this is the cell. This is the internal environment of the cell, which is the cytoplasm. This is the external environment, which is ECF. And this is the phospholipid bilayer. In case of neurons, all of this is true. All of this is true, but a very important thing, the three regions of the cell neuron, three regions. One, the dendrites. Second, the cell body. Third, the axon. And fourth, actually, the axonal terminal. Each of them have a slightly different composition of the plasma membrane. Why? Because each of these regions have different functions. So it's very important to understand that the plasma membrane of the neuron at different region have a different composition. The plasma membrane of a neuron is structurally and functionally specialized in different regions to support efficient signal reception, integration, and transmission. Very important to understand again. Signal reception, integration, and transmission. In the dendrites, for example, in the dendrites, as you can see in this picture, the plasma membrane is highly enriched with the ligand-gated ion channels and receptor proteins, allowing it to receive, because it's a receiving end, and respond to neurotransmitter at synapses. Structurally, it often forms dendritic spine that increase the surface area for synaptic control. We learned that, right? It has synaptic spines that increases in number, and these ligand-gated ion channels that are present in there help as a receiving end of the neurotransmitters. The second part, the cell body in here. The cell body or soma membrane contains high density of leak channels. This is new. This word, leak channels, is new now. I'm introducing it, and you will see that we will learn more about it in action potential when we learn. These are also channels. These are made up of protein. These give free transmission or free inflow of sodium and potassium ion. These are leak channels. There are two main leak channels. One is sodium leak channel, the other is potassium leak channels. They are highly concentrated in the soma. They help to supporting metabolic activity and integration of the incoming signals rather than rapid electrical conduction. Then the next region is the axon hillock, which is a trigger zone. At the axon hillock, an initial segment, the plasma membrane is uniquely specialized with exceptional high concentration of voltage-gated sodium and potassium channels. Why? Making this region the trigger zone for action potential. This is the region. This is the region where the action potential is initiated. So, this axon hillock region has very, very high concentration of voltage-gated sodium and potassium channels. The last region, which is axon, not the last, we have the last region as axon terminal. The axon, the membrane is adapted for rapid impulse conduction with evenly distributed voltage-gated channels in unmyelinated axons and concentrated at nodes of Ranvier in myelinated axons. So if these are, for example, this neuron shows this is myelinated. So this, if it is myelinated, the nodes of Ranvier where the myelination is not there, it is highly, highly concentration with voltage-gated channels. And if it is not myelinated, it is evenly distributed. These gaps in between the myelinations are known as the nodes of Ranvier. Finally, the axonal terminal. The axonal terminal plasma membrane is rich in voltage-gated calcium channels. So this calcium now plays a very important part in the diffusion or release of the neurotransmitters that are present in here. If you look into this picture, you'll see this is the axon terminal. This is the postsynaptic neuron. So these vesicles diffuse and come into this region. And this process of diffusion through the membrane is only possible with the help of calcium channels. So calcium, the level of calcium channels is highly concentrated in the axonal terminal. This is a very important function of the axon because the plasma membrane at the different regions of the plasma of the segments of the neuron is different and is highly specialized for its different functions. The cytoplasm, the neuron, the cytoplasm, which we are talking about, the soma, has pretty much the same content like any other cell. It is mainly responsible for protein synthesis. We learned that the proteins, there are several proteins that are required to be made by the soma, which can be neurotransmitters, it can be enzymes, it can be sodium potassium channels, it can be leak channels. All those are made up of proteins. So these have to be made in the soma with a blueprint of DNA in the nucleus and following the pathway of transcription and translation to form the proteins. Cytoskeleton, we learned about that, how these actin, microfilament, neurofilament, microtubules are important in the neuronal structure. There are 3 different bones in the neuron, which are microtubules, microfilaments, and neurofilaments. All of them have important function. Disarray of it leads to Alzheimer's disease. Ribosomes, as all new cells have, these are the building blocks of proteins, site for protein synthesis. Ribosomes and rough endoplasmic reticulum both help in the process of protein synthesis. Mitochondria, this is the powerhouse of the cell. Remember, neurons are highly active, and in order to do its function, the mitochondria is highly evolved, has very similar function, similar structure as any mitochondria in the other cells, and it contains its own DNA and RNA. ATP is highly required, and remember, one thing to remember in here is glucose is the only source of energy in case of neurons. Endoplasmic reticulum, or rough endoplasmic reticulum, in case of neurons, is known as Nissl bodies, which helps in the protein synthesis. Once the protein is made, it is packaged into the Golgi apparatus, and then it moves out of the soma to the different regions where it is regulated. So the axonal structure and functions, we did that. This is the axon hillock. Axon, these are collateral in order to increase the surface area. We learned that in this region, there are tons of sodium-potassium pumps, which is the trigger zone, which causes the action potential, initiation of the action potential. And once it's initiated, it passes through the axon and goes into the axon and terminal, synaptic terminal. It is also known as, here there are vesicles which carry the neurotransmitters, and these, with the help of calcium, are released into the synaptic cleft, which is taken up by the postsynaptic neuron. Important information, the neuron does not have a centriole. We have discussed that, and hence does not undergo cellular division. And the axonal transport, very important. The proteins that are made in the soma, we learned about that. That needs to be transported to the axonal terminal. And in the axonal terminal, certain proteins or degraded proteins or toxins need to be transported back to the nucleus. In order to do that, there are railway tracks present in the axons, which are stabilized by the microtubule. And we just learned, like, if these microtubules are non-functional, what they cause is deposition of tau proteins, which can lead to Alzheimer's disease. These rails, as you can see, there are two types. One is known as anterograde, the other is known as retrograde transport. Anterograde transport means going from the cell body to the synaptic terminal, and retrograde means moving from the axonal terminal to the cell body. So there are railway tracks and there are rails in there which does the work. The anterograde is done by kinesin protein, and the retrograde is done by the dynein protein. So these are the railway tracks. These are the train compartments, you can say, which carry the information, and these are the ones that carry information from the synaptic terminal to the cell body. The one that helps in anterograde is kinesin. The one which helps in retrograde is dynein. So the direction, anterograde and retrograde transport. So if you look in here, anterograde transport from soma to axonal terminal. In retrograde transport is from axonal terminal to soma, reverse. The protein involved is kinesin. This is dynein. The speed can be fast, it can be slow. This is always fast. What is required? These are usually neurotransmitter, mitochondria proteins. These are debris, old vesicles, growth factors that need to be either discarded or recycled. The role is supplies to synapse, and this is mainly for recycling. Pathogens, nothing goes from anterograde, but there are certain diseases and certain toxins. The retrograde transport is required, but in the process, retrograde transport also causes certain pathogens to be transported from the axonal terminal to the cell body, which can damage the neurons. Axonal terminal, as we learned, important thing to remember, it does not have microtubules, the terminal, but it has plenty of synaptic vesicles. And these synaptic vesicles are filled with neurotransmitters, which are made in the soma. There are abundance of membrane proteins and large number of mitochondria because it is an active region where uh, the neurotransmitters are released from the synaptic terminal into the synaptic cleft. So as you can see in here, we'll learn more about synapse later on, but this is the presynaptic neuron with the vesicles. These are the mitochondria that help in the process, and these neurotransmitters are being discharged or released into the synaptic cleft, and the postsynaptic dendrite is taking up these neurotransmitters. And the postsynaptic receptors are present on the surface of the dendrites, which can take up these neurotransmitters. There are several diseases associated with it. One is amyotrophic lateral sclerosis, Alzheimer's disease, Parkinson's disease, Huntington disease, all associated with the axonal transport defects. With this, I conclude the first part of the lecture, which is neuron. The second part of the lecture is on neuroglial cells, the supporting cells. I hope you understood the neuronal structure, the function, and the importance of neuron in the nervous system and in whole, the entire body. Thank you.